my pleasure to introduce Sabine Kastner, who surprisingly and incredibly grew up in Hanover, Germany. And let's see, parents are still there, what else do I know? You went to Goethe as an undergrad and studied history and philosophy. And then Goethe, you went to London to do your neurology. So she's an MD and a PhD. And then went to Goethe again to her postdoc. Then did a second postdoc with Leslie Ungerleider at NIH for 4 years. And since that time has been at Princeton. So let's welcome her.
Thanks, Peter, and thanks for having me. I had a really wonderful day and lots of very inspiring discussions with you, or with some of you. So I would like to tell you today some of the electrophysiology work that we have pursued in my lab during the last 5, 6, 7 years or so. And I would like to start my talk by introducing you to my favorite music. And also to tell you some of the basic facts or things that we study in the field of attention. So basically what this kind of scene shares with other scenes in our natural environments is that it is extremely cluttered and contains many different objects. And you may think that intuitively and subjectively you take all that information in and you can represent it, and even if I would remove that scene, you could tell me a lot about the details in that scene, more so because you know the scene probably, or some of you, relatively well. And I want to show you that this is actually not true. So what I'm going to do is I will turn this into a little experiment. There will be 2 frames of the same scene here, and there's a small change introduced. And I would like you to show me with a hand sign, don't give it away verbally, what the change is. Now let's see if this works. So some of you got it right away. It's not a very difficult change. Who didn't get it yet? Okay, so there are a few people, that's good. So basically I'll let you just look at that for a little while. What little experiments like that show you is that the capacity to process visual information is extremely limited. And this is actually why we need mechanisms for selecting behaviorally relevant information. And that is how I want to define attention today. And this has hiccuped probably a few times. So what we simply did here was to provide Paul McCartney with a pair of shoes, because he is famously barefoot in this original scene here from the Abbey Road album. And I want to give you a little bit of an overview today of what we have learned about the mechanisms underlying these sort of selection processes. And the selection that I'm going to highlight today is what I try to emphasize here. So once I pointed you, or those of you who did not make that detection of the change yet to that location, you were easily able to overcome your limited processing capacity and to detect that change right away. And this is what we call a space-based selection. So this is guided by a cue in space. And I will just constrain my talk today to these kind of space-based selections. You can select based on many, many other criteria. You can look for white things in the world, or for categories in the world, for people or cars, but I will just keep it very simple today in my talk. So when we look in an imaging experiment at the typical network activity that we observe during a space-based selection, this is something that we would get. This is from a human brain imaging experiment. So this is a right hemisphere. It is inflated and very distorted. This is the frontal pole, and this is the posterior pole. And what you see here animated is the typical fMRI activity that we would get in a spatial attention experiment. So you see these kind of representations here in the visual system that build up first. And then with a little delay, we get activations along the interparietal sulcus here in the posterior parietal cortex. This is this region. And we will get frontal activations that cannot be appreciated in this view very well. And what these kind of frontoparietal activations do is simply sending some signals, we know that from other experiments, down here into the visual system to stabilize the ongoing visual representations in some way. Now, from what I find interesting with these sort of network activations that we have seen not only in the framework of attention but in many other contexts of cognition is that we get a really large-scale network engaged, and within a 300 or 400 millisecond frame, we get a behavioral response. And one of the questions that I have become very interested in is how the communication operates in these kind of large-scale networks and how we can get at that question. But let me just tell you how, if I can move this forward, how we basically, in a textbook-like fashion, conceptualize attention and attentional selection at this moment in time. So we assume that there is some information coming from the retina going to the thalamus and then being represented in multiple visual areas along this two-stream model here, and then there is some higher order activity in prefrontal and parietal cortex that generates signals that then feed back into these visual representations and somehow modulate the ongoing activity in visual cortex. So that's kind of our standard model of top-down effects that a cognitive process may have on sensory processing. And I want to show you today, first of all, this is a really strong oversimplification, and we need to modify this scheme quite a bit. And the two broad questions that I would like to discuss are here. So the first one in the first part of my talk will be how is communication set up in large-scale cognitive networks. So that's just a question that I basically introduced you to. And the second one, in the second part, will be how which neural code can predict behavior. So how can we define neural signals that can be read out to drive behavior? So these are the kind of very big questions that we try to get at, and I will show you some first evidence today. Now, we study these kinds of issues of mechanisms for attentional selection and mechanisms for network communication in two different brain models in which we use a set of different methods. So this is the human brain model over here, and this is the macaque monkey brain. And in both of these brain models, we do a lot of different functional imaging. We do functional magnetic resonance imaging that I've illustrated to you. We also do diffusion tensor imaging to define the network connectivity in greater detail, and so on. I will not talk a lot about that today. So today I will mainly talk about the electrophysiology, but I want to mention the imaging because it really guides the electrophysiology, so one cannot live without the other very easily. And I will introduce you to both of these sets of studies that we do. So we do recording simultaneously from multiple sites in the monkey brain, a monkey that is trained on a relatively complex attention task. And then we do the same experiments, so the same attention experiment in human patients that will undergo epilepsy surgery and are temporarily implanted with electrodes, where we also have the opportunity to do intracranial recordings, and I will introduce you to that in the second part, and I do that together with Bob Knight at Berkeley. So let's start with the first part. So this will be work on non-human primates exclusively. So the question here is, how is communication set up in large-scale cognitive networks? And the issue that we were particularly interested in this particular set of studies was what kind of role the thalamus plays in setting up cortical network interactions. So the studies were done by three really brilliant scientists, a very, very good electrophysiologist, Dewey Salmon, who has now his own laboratory in Wisconsin, Liang Wang, who is an applied mathematician and who helped a lot with the data analysis of these rather complex set of data that I will introduce you to. He has now his own laboratory in Beijing, and my long-term collaborator, Mark Pinsk, who did all the imaging that really helped us or were the key to doing the studies that we did here. Now, I give you basically the take-home message of this part of my talk up front, because it has happened in the past that I'm losing people during this part, and so even if I lose you at some point, you will still have the take-home message, so I want to make sure that you get that. What I try to show you is some evidence that the pulvinar, which is a large nucleus in the thalamus, plays a very important role in setting up the communication in this large-scale network that I've introduced you to, the large-scale network for space-based selections in time. So it basically makes the communication in this network efficient by aligning the network in time so that these different nodes can talk to each other. So that's what I will try to show you some evidence for. And if you lose me or I lose you on the way, then we at least can share this important conclusion. Now let me introduce you to the thalamus. That's one too far. So here is a sagittal slice, or a series of 3 sagittal slices of the monkey brain, and the thalamus is this region here that's buried under the cortex. And in a schematic, this is how it looks like. And the 2 visual nuclei of the thalamus are a structure that you know here very well, Surfan's work, the lateral geniculate nucleus. This is the relay nucleus that transmit the information from the retina to a primary visual cortex. And then there's another large nucleus that's over here. This is actually the greatest nucleus in the thalamus. That's the pulvinar. Now, the pulvinar has quite some different characteristics as compared to the LGN. So as I said, the LGN gets a significant input from the periphery, from the sensory periphery, from the retina that gets conveyed to the cortex. The pulvinar, on the other hand, is almost exclusively interconnected with the cortex, so it does not get a major input from the sensory periphery. Now, we can look at these pathways a little bit more closely. So what I show you here are 2 areas that are 2 cortical areas along the ventral stream, area V4 and TEO. So these are areas that are form and shape selective in the primate. And this is the 6 layers of cortex over here. And the way the input-output connections work is that, first of all, these 2 areas, and that's very, very important for our context, have a direct cortical-cortical connectivity, that is this area over here. And it is an anatomical rule in the primate brain that any 2 areas that are directly connected in the visual system at the cortical level will have an indirect loop that goes through the pulvinar. And that is depicted here. So this will take off here from a layer 5, and then we'll go into the input layers of the next area. And since this is an anatomical rule, so that is true for any 2 areas that have this direct cortical-cortical projection here, there should be some kind of special functional role, but there's basically nothing known about it. So we wanted to get at least some kind of functional role for these indirect loops that the cortex has with the thalamus in this particular study. Now, we thought this is very straightforward. What we did was we wanted to record from 2 cortically connected regions. So we took these 2 along the ventral pathway, V4 and TEO. We also knew that both of these regions are very nicely modulated by attention. That was very well established in the literature before we started these studies. And then we wanted to basically identify the projection zone that these 2 regions share in the pulvinar, and then train an animal on an attention task and do these 3-way simultaneous recordings. And I used to have another slide in my talk that pointed to the exact methods, how we identify these projections. This is all done with imaging. But I took it out because it's too frustrating in a sense. It's 4 years of work that went into one slide. And so 4 years later, we were then ready to set out to do these studies. But it was methodologically quite challenging to actually establish these simultaneous recordings. Okay, so we tested a very specific hypothesis in this study, and I want to introduce that first before I show you the task that we used to probe this. And I want to also tell you a little bit about how we derived at this hypothesis so that you understand that better. So our hypothesis was that the pulvinar may synchronize neural activity in two interconnected cortical areas in order to optimize information transmission between them. So we drew on a number of findings here. The first finding is that spiking activity in any given area here that we can think of is often not so spontaneous, but there are regularities to it. So here, for instance, we see an oscillation in this area that we can measure in the local field potential. So that's basically a local neuropopulation signal that we can pick up. And the interesting thing is that spiking activity is more often related to certain phases of an underlying oscillation. So here, for instance, it would be the peaks of that particular oscillation here in this example. Now, if two areas want to communicate with each other, it's quite good for them to actually get into synchrony with their oscillation activity, because then a spike that travels from area 1 to area 2 can basically can be conveyed, because it will meet this kind of excitable window here, and not a situation where these areas are out of sync. And basically, this spike here would meet a relatively inexcitable window. So that is one observation that we built this hypothesis on. Now, then you can think about, and yeah, I should mention that there was some very, very good evidence in the literature at the time when we started these studies that these kinds of oscillatory patterns or patterns of synchronous activity that two areas try to set up, here these are the frontal eye fields in area V4, that cognitive tasks like attention can influence these oscillatory patterns of activity. So we knew that cognition can at least potentially influence this. Now, then you think about the question, okay, if you have these two areas and they want to now start to talk to each other, so they want to set up some kind of synchronous activity pattern, how can they do that? So one way is that they can use their feed-forward and feedback connectivity that they share to set this up. They can go back and forth and negotiate whatever a good communication state or state of synchrony is for them. And the alternative hypothesis, this is what we tested here, is that actually a third area is doing that for them that is interconnected to both. So this, in this particular hypothesis, is of course the pulvinar that has this loop that could basically control this kind of timing in these two regions here. So this is basically where this hypothesis that the pulvinar may synchronize neuroactivity in two interconnected cortical areas came from. Now, by the way, if you have questions, just ask them right away, okay? Don't wait until the end until you have forgotten them. So let me now introduce you to the tasks that we have used in these studies and that we are also still actively studying and probably for many years to come. So when I started to go back and do some electrophysiology, I thought one thing I wanted to do a little bit different than what is done classically in my field is that I wanted to use tasks that are inspired from human cognitive psychology and lead to a little bit richer behavior than what often is done in monkey physiology studies. In the first task that we set out to study, we have now a second attention task that we set up in a similar way going, but I'll talk about that probably next time when I come, is actually the most studied attention task in the entire literature. That's the Erickson flanker task. Hundreds of publications in the human behavioral literature since it was first introduced in the 70s. And typically this task is done with letters in humans, and we simply chose some more neutral stimuli for our animals, and we just replaced the letters by shape stimuli. So this is how this task goes. We present a fixation point. The animal will always maintain fixation. There's never an eye movement involved here. So all the attention operations that are involved here are covertly without the execution of eye movements. Then we show very briefly a cue in one of 6 or 8 locations in the periphery. They are arranged in a circular manner around fixation, as you will see in a moment. And we basically ask the animal to maintain spatial attention at that location, to memorize this location. And then there's a longer delay period that can last up to a second. And then there is an array that's made out of these 2 different stimuli, bow ties and barrels, that we present. And then the animal responds with a manual response, whether there is a bow tie or a barrel stimulus in the cued location. So it's a very straightforward task. The nice thing, and this is why it's called a flanker task, that you can introduce here, is that leads to different behavioral outcome, is that the target stimulus can be flanked by either non-congruent stimuli, like in this condition, or by identical stimuli, congruent stimuli. And this leads to different reaction times. And I'll show you that here. So this is the monkey behavior. This is reaction time. These are the 2 different flanker conditions here, the congruent and the incongruent. And you can see that you get a little bit of a reaction time caused in the incongruent condition, so it takes a little bit longer to respond. And the size of this effect also depends on the overall number of distractors in the display. And this is what's called a flanker effect. So this is where that name comes from. And here's in comparison the human behavior, which again has been shown and replicated in hundreds of studies. It's a very robust design. And of course, we see here the same flanker effect. So that was a nice thing, because here we establish that monkeys have a flanker effect as well. And the nice thing for the monkey physiologists in the room, or people who are familiar with that work, is that this is so robust that you see it after thousands of trials. So it's one of these effects that do not go away, which is very nice. Yeah?
What accounts for the difference in overall magnitude between monkeys and humans? So the monkeys is doing it lots more times, so they're faster?
They're faster. I think they're also more motivated, but I don't want to insult our human subjects here. But that's my personal interpretation, so just take it as that. Well, you know, monkeys are also smaller. You can also see that just simply as a problem of, you know, the information has to go a longer pathway to the effector, but there may be many reasons for that. Okay. So we had some hope when we started these studies. Why would you go to the pulvinar? You know, why not to some other structure in the thalamus? We had some hope that we would actually find some responses there that would get modulated by attention. And this builds on work, on very early work that was unfortunately left behind in the 80s, relatively soon, by Steve Peterson, who had done recordings in the pulvinar and had found modulations. I don't want to put you through the details of this slide, but basically, once the field moves to the awake behaving preparations in non-human primates, the entire field goes relatively quickly to the cortex and then forgets about the thalamus for reasons that I never understood, but it's just a matter of fact, as you know, you can attest it as well. So we have these early data, but they were never really followed up upon until, you know, relatively recently, and there were a few laboratories, not only ours. So I want to show you first just single unit activity from the pulvinar, just to make the point that the pulvinar really is engaged in this task, is doing something interesting. And the way I will present also in the following slides this this activity is that we will look at activity at a receptor field that we're recording from when we instruct the animal to attend to that receptor field. That would be what we call here the attention in condition, this red trace. I explain what's going on in a moment. And we compare that with this blue trace of activity here to a condition when the animal attends to the opposite location, to the receptor field we are recording from. This is the attention out condition. So that's basically our control condition, if you want so. So what's going on here? So we get at this point 0 here a nice Q-evoked response that then dies down. And then we have here a variable delay, so the delay is not predictable for the animal. Excuse me, it could be anything between 300 and 1,000 milliseconds. This is why this is broken up here. But you see very clearly that towards the onset of the array, which would be the second 0 point here, you get a very nice elevated delay activity. And then you get a moderate modulation also of the target evoked or array evoked response. In the following analysis, we will focus very much on this elevated delay activity because here we have a stationary period that is basically a pure cognitive state. It's an attentional state. There is no input from the sensory environment. The cue has been switched off. It's not affecting the neuroactivity anymore, and the animal basically waits for the array to be switched on. So that will be our main focus here. Now we go and want to look at what happens in these three-way recordings and how can we now look at different oscillatory activity in these three different regions and states of synchronous activity and how it changes depending on where the animal attends. Now the signals that we will look at now are local field potentials. So these are local EEG signals, if you want so, from small population of neurons. And we have made sure that these populations of neurons are interconnected. Probably not one-on-one, but they can be indirectly connected. So we don't care too much about that. They share receptor fields, and as you will see, they also this is also reflected in these synchronous activity that we can measure. Now, the first question that we looked at is what happens, and these are pairwise comparisons, what happens with the two cortical regions when the animal attends to the receptor field we are recording from, or when the animal attends away from these regions. What happens to oscillatory activity? Does it get modulated? We expected that based on earlier work from Earl Miller's lab, from Bob Desimon's lab, Pascal Free's lab, and so on. And this is indeed the case. You see there's an increase here in synchrony of oscillatory activity in the alpha frequency band. So that's a band that peaks around 10 hertz when the animal attends to the receptor field. And this is for V4 and TO. Now, the next thing we looked at was what happens now if we look at the pulvinar in one of these cortical regions. And what you see here is pretty much a similar increase, again, in this frequency band that peaks around 10 hertz. And there's an increase in synchrony. And again, this is all during the delay period when there is an attentional state and no further sensory input to the system. And we would see a similar pattern, I just didn't print it here, if we looked at the synchrony between the pulvinar and TO, the second area. So what that basically tells us up to this moment in time is that these three regions indeed are all engaged in this task. So we see modulations by attention of local field potential activity, on oscillatory activity, on single cell activity, and so on. And it seems that basically this mini network that we have carved out here uses this 10 hertz frequency band to communicate with each other. But then the question is, what is the directionality of this communication? So is this coming from the cortex and going to the thalamus, or from the thalamus to the cortex? Those are the real interesting questions that you would like to get at once you have established a little network like this. And we have not done the experiments that we really need to do, which would be lesion experiments. So basically just knocking out one of these nodes here and see how it affects the other two nodes that you're recording from. But we have tried in the first approximation at least to form a hypothesis about this information flow. And the technique that we used is a statistical technique. It's Granger causality. And this is the idea of this method. It is built upon temporal precedence, so the idea is relatively straightforward. You see two different patterns of activity in two different regions. The blue one would be the pulvinar here, just hypothetically. The red one would belong to TEO. And the pulvinar pattern precedes the pattern that you then measure in TEO. So we would think that the information went from here to there, not from there to here, just based, again, on this temporal precedence assumption. Now, you can do these analyses. Again, these are pairwise analyses that you do, but you can do them conditionally by taking into account that there is a third player, at least a third player, in this network. In this case, we have these data from before, and that's what we call a conditional Granger analysis. Now, when we do this analysis, what we observe is basically that the pulvinar, here's the Granger causality of the pulvinar and V4, again, under the condition that there is a third player, that there are data from TEO. And what you can see is that there is this increase in Granger causality that the pulvinar has on the cortex. And again, it is in this kind of 10 Hertz frequency band. And when we look at the second set here, that's the influence that the pulvinar has on TEO, you see again that there's this increase in Granger causality, in the causal influence that the thalamus has on the cortex. Now we thought that was interesting, but then we thought, okay, what is the cortex doing? It has also, this is the indirect loop basically that could be used in order to set up this pattern of directionality of information flow. But the cortex has also feed-forward and feedback connectivity. So what is the cortex doing with that connectivity? Does it have some kind of influences upon each other? And the kind of shocking finding was here that the cortex itself does not get modulated. So here, if you look at the Granger causality, the causal influences of V4 to TEO, given pulvinar, there's no modulation whatsoever, so that would be the feed-forward connectivity that could be used to modulate. And if you look at the reverse one, the influence that TEO may have on V4, given that there's the pulvinar, again, there's no modulation whatsoever, so also this feedback connectivity that these areas have does not seem to get used here. So it seems that during that delay period, when this cortical network basically gets set up and aligns in time to get prepared to make this selection, all the information that gets conveyed comes from the thalamus and goes to the cortex. And our interpretation is, again, that this alignment in the alpha frequency band is used to basically temporally structure this network. We have just looked at regions here along the ventral pathway, but we have some evidence that this also actually relates to the higher order network and that the pulvinar is doing similar things there. So it could be that basically it's just a structure that provides this kind of temporal context that the cortex needs to operate on. So let me just summarize this part of my talk by saying that the pulvinar regulates synchrony between cortical areas according to attentional demands by influencing the cortex in the alpha band. In this particular task, so don't generalize it easily to others. Other tasks may have other, or may use other frequency bands for communication, but in this task, that's what we found. And by regulating cortical synchrony, the pulvinar may control information transmission across the cortical attention network. And we think more generally that we should consider the pulvinar as a subcortical control structure, a kind of clock for the attention network. It's very possible that the pulvinar plays a role in many, many other cognitive tasks. And I think it's a good time for us to rethink a little bit our simplified versions of what top-down is, that something comes from prefrontal cortex and gets transmitted to sensory processing. I think this example shows us that we have here a much more complex scenario that we need to consider. Okay, so that brings me to my second part. And in that second part, I want to show you some data from the same task. So the task will not change, but I want to show you also some data from human ECOG recordings. And the leading question that we had in this particular study is, which neurocode can predict behavior? And the collaborators stayed mainly the same, but we had one new one, and this is Bob Knight, who really was one of the first who did electrocorticography in humans and has set up a wonderful network of colleagues to do this. I know that you do electrocorticography here, so probably I don't have to really introduce you to it a lot, but I just briefly want to do that. The patients that we have the opportunity to record from undergo neurosurgery. They are all epilepsy patients, at least in this cohort. And in preparation for the neurosurgery, they get implanted a week before the surgery happens, with grids like this. It gives you just some examples. These are intraoperative views, and this is just how these grids end up on the cortex here in 4 patients. For diagnostic purposes. So basically what's done here based on the individual case history is to implant these patients and then to test the functionality of the underlying tissue and to identify the epileptic focus very precisely, and so on and so forth. And since these patients are on intensive care and often very bored, they sometimes agree to get involved in our also probably quite boring experiments, and then we have the opportunity to get really, really unique data. But an important point to make here is that these grids are not implanted for any scientific purpose. They are only implanted for diagnostic purposes, so we as scientists never have an influence on that. And you may wonder, why do they have so much, parietal coverage there? Because that's really one of the regions that interest us here in the study, as you will see. The way we operate in this consortium of scientists is that the study that has an interest in parietal cortex would get preference for a patient who gets implanted over parietal cortex. So we basically select very carefully the studies that we do depending on the coverage that we have in these patients, okay? So in individual patients, you may get very variable coverage, but if you put these patients then together in a larger cohort, and here I just have 5 patients put together in these different views, we have 8 now in the study. You actually get really, really nice coverage. So we have 800 electrodes in one hemisphere from these 8 patients. And these are 3 different views. So this is a medial view, a lateral view, and this is a posterior view here on the visual system. And when I started these recordings, one thing that really bothered me was that I never knew where these electrodes were. I mean, it's kind of like you have Thalamus coordinates or MNI coordinates or something like that, but that doesn't really tell you much. So I thought probably we can use some of the imaging data that we have acquired for more than a decade and can make more sense of that. And what we did, and this is now online, and anybody else who's interested in that can download that, is a really nice tool, is we have developed a probabilistic atlas of the human visual system. So the human visual system has about 30 areas cortically, and you can delineate them. The main criterion is here representation of visual space, or portion of visual space. And we have put 53 data sets where we mapped out the cortical visual system in this way. from our imaging database. We put this together and derived a probabilistic atlas in a normalized space. So if you have, a patient data set, or if you have an imaging data set, but you cannot do retinal topic mapping, you can just basically load it in there and you get a better idea where your activity falls. Or here we see where these electrodes fall. So here, for instance, we can say with a great or a high probability that these are V1 electrodes and so on. So it gets us a little bit further and a little bit more confident where the electrodes of interest for us fall. Now the region, yeah.
So online, is there like tools to put in Colorado coordinates and then you get image?
It basically interacts with all the major platforms. So with FSL, with AFNI Summa, so with the major imaging platforms that you would use for imaging analysis. So you can interact and load it into those platforms. Yeah. If you have thoughts about how we can develop this further or improve it, just let us know. So that's kind of ongoing service for the field. So the region that we were particularly interested in this particular study is the lateral interparietal cortex, or the posterior parietal cortex in humans and monkeys. So this is the lateral interparietal area in monkeys that we recorded from, again, single unit activity and local field potentials in this Erickson flanker task. I just want to remind you briefly of some of the characteristics of neurons in this area. So these neurons have spatially confined receptor fields. This area has very strong structural connections to the frontal eye field, and it is part of a circadian attention network, and LIP has also been involved in other cognitive tasks that were not so much of interest here. So when we started these studies, we sent a grant to NIH. It's always a good starting point, as you know. And our review said, well, I don't know what LIP is in the human cortex, so you have to make a case what you're comparing here. We just said we would go after the posterior parietal cortex. We were a little bit hand-wavy with that. And they were actually really right. This is a very, very important and very difficult problem to solve, to really track down which area in the monkey brain you can compare, especially in higher order cortex, to the human cortex. So I want to go into this just to kind of give you the flavor and the size of the problem by just showing you how the interparietal sulcus in the monkey is organized. So here we have this lateral interparietal area, which is basically linked to eye movements as effectors. I have grouped this by effectors. Then we have another region here, the anterior interparietal region, which is a grasping area. And then we have another one over here, this green region on the other bank of the sulcus, the parietal reach region, where you have reaching movements represented. So let's just look how this all looks in the human. So what we see here is really different. I should also make the point that the only region that has a spatial map is part of LIP. So all these other regions would not have any specific spatial organization. In the human, and this is again this bird view, this is a posterior pole, this is a frontal pole, and this is the open interparietal sulcus, you have 6 topographic regions along the interparietal sulcus. The reach region is dislocated into the superior parietal lobule. The grasping region is actually falling onto the intersection with the inferior parietal lobule, so also basically dislocated outside the sulcus. And you have now this problem, okay, which of these regions is LIP? So we had to convince ourselves that some of these regions are more LIP-like than others. And I will make this case today that we think these 3 regions in the human have LIP-like functionality or characteristics. And I'll tell you why. So we have a few arguments to make. One is, and I think this is the single most important one, these 3 regions are exclusively connected to the frontal eye fields. The others are not. So here's the connectivity in diffusion tensor imaging case of frontal eye fields and these 3 regions in the posterior parietal cortex. And then we see in these regions also a number of response characteristics that we can measure in normal subjects that are LIP-like. So we see a strong preference for saccadic eye movements over smooth pursuit eye movements, for instance, very strong attention signals, very strong motion representation of different motion stimuli, and also a strong preference for shape stimuli. That's also very characteristic for LIP in the monkey. So these are basically the kind of data and arguments you can make to argue that, you know, you can compare those two. This is probably how far you can push it just based on what we have in the human at this moment in time. So you can believe me or you can say, okay, go away. Our reviewers believed us, so we got the grant funded. That was good. So I want to show you also something nice now from the atlas. This is again a different view of this interparietal sulcus. These are two electrodes that we will look at in one of these regions, interparietal sulcus area 3, so we number them simply, and I will show you two electrodes from outside. So again, these are all have representations of space. It's basically the contralateral hemifield. And the nice thing is that we can measure very nice response fields that just look like receptive fields. People often say, oh, ECOG data, they are so unspecific. They're not. They're very specific if you ask specific questions. So here you see this blue electrode here that has a response field along the horizontal meridian. It's very confined. And the red electrode over here has this response field along the vertical meridian. And then the two electrodes outside, this topographic structure, they don't have much of a response field, so they are kind of nice controls. And this is really nice because now you can start asking questions just like in the monkey, what happens when our patient attends here versus here or here versus here. So we can ask very similar questions, just looking at different signals if you want so. I will introduce you to those in a moment. So let's just look at the lateral interparietal area and what goes on in our paradigm. It looks very much like the pulvinar, so qualitatively not much difference here. Q-evoked responses, these are 50 single cells that we pulled together here, elevated delay activity, and then a moderate attentional response enhancement. So this region is non-surprisingly involved in this task, of course. So let's just look at one of our ECOG electrodes to see how this compares. So here what I show you is this is the time relative to Q onset, and this is the time relative to array onset. So this would be pure delay activity. And these data, these are complex data, and you have to break them up into different frequency bands. So here we look at frequency bands from, you know, where our cutoff is, it's about 4 hertz, all the way to almost 200 hertz, okay? And the major signal that you measure here is in the major power of the signal is in the frequency bands between 80 and 180 hertz. So that's what we call the, you know, broadband response or high gamma activity. You can think about this high gamma activity as spiking activity in an unsynchronized fashion. So it's basically like a bubble of, you know, very fast spiking activity that's uncoordinated. And we see here very nicely that we get a strong onset response that's somehow related to the Q, but of course we cannot make out where it ends. We have a longer delay period here for our patients. It goes up to 1.3 seconds. So these are the last 300 milliseconds before the onset of the array. And you see here very, very beautifully delay activity when the patient attends to the response field and when the patient attends away from it. So there's a very strong difference here. And then this is the array-evoked activity. I show you, I think, some modulation of that later. So the first question, this is kind of how it compares. So we see, you know, similar signatures here, but they lay in a very, or they are in a very different format, if you want so. But there are some qualitative similarities. So the first question we asked is, okay, what does this high gamma activity mean? What is it in terms of the signals that we can measure in the monkey? What is it most closely related to? So what we did was we took our local field potential data and broke it up into different frequency bands, and then we correlated the spiking activity with these different frequency bands, which go here from low theta, that's 4 to 8 hertz, all the way to the traditional gamma, which is 40 to 60 hertz, and then high gamma, which is everything above 70 hertz here in our case. You can see that spiking activity correlates best with high gamma, and gamma activity so with the faster frequencies. And from here on, since we will basically just look at high gamma activity in our patients, I will basically treat the signal as a surrogate of spiking activity that we can measure in these ECOG data. Yeah?
Was the correlation between spiking done on human data?
No, these are LFP data because we don't have spiking data in the human data set. So there we just get ECOG data, which is basically, a very broad EEG signal that comes from probably 10,000 underlying neurons or something like that. So it's a surface electrode that picks up these signals. These are monkey data. This is LFP. Sorry, this is the spike activity in the LIP recordings that I showed you, and the local field potential. So we just filter the local field potential into the frequency bands and then correlate it with the spiking activity that we have measured. Okay. So the question that we wanted to ask here is, so we have now all these signals. We have these high gamma activity in the human ECOG recordings. We have single unit activity. We have oscillatory activity and whatnot. So the question was for us, which of these signals can best inform us about the behavioral outcome? And what we considered here were two different codes that you may, or that the brain may take. There may be many, many more that may get implemented, but we just probed these two. And the first one is the most traditional one. It's a spike rate code, which simply sums up or averages, spiking activity in a certain time window. Here you would have 2, and in this time window 1 and 2, and so on and so forth. But this code does not consider that sometimes spiking activity occurs at different points in time. So in that respect, these two spikes are quite different from these two spikes, right? Though in this code, this timing information is thrown out, so they both end up with the same count. So if you want to take into consideration that that spiking could be tied to certain time, then you can link the spiking. Again, this would be the same examples as here, to different phases of an underlying ongoing oscillation that you can measure on the local field potential. So schematically here, you would now end up with a very unique code. So you would have here zeros for these particular phases. Then you would have one spiking event, another spiking event in these two phases, and then another 0 here in this red phase. And you would end up with 3 very unique codes for these 3 different examples that I give you here. So this would be a spike phase code that we considered here as a second possibility. Now let me show you first that both of these codes are actually implemented in the lateral interparietal area and also in the posterior parietal cortex in these 2 primate brains. So I'll show you the example first for the spike rate code. And I've given you many examples before in this talk about that the spike rate code really is implemented in this paradigm. But here's yet another example. So what we're looking at here are 4 different locations that the animal's attention is directed to. 2 of them, P2 and P3, fall into this receptive field that's schematically shown here in blue. And these P1 is at the border and P4 is a little bit outside. And we look at these 4 locations here relative to the cue and during the delay period. So this is a delay period, and this is the cue-evoked activity. So let's just look at this one over here. So you can easily see that the rate, the different rates here during the delay period informs us of where the animal attends, right? So this green rate, which is the highest, tells us that the animal attends to P2. That's the peak of the receptive field. the blue rate tells us that it attends to P3, and so on and so forth. So let's see if there's also a spike phase code here. So here I show you from the same neuron, this is just one example, single trials of the oscillation that's the strongest here during the delay. It's a beta oscillation that peaks at 17 hertz. At P2, again, that's the peak of the receptor field, and here these are the spiking events in these single trials, and we coded them just schematically here to different phases of this beta oscillation. And you tell me if you think that one spiking event is more frequent than another one by this color code. Can I get a vote on or guess on something? Green or blue? It's noisy, yeah, but they are a little bit more frequent, certainly, than red, for instance. So now we look at P3, and what do you think about P3? Is there a more frequent phase spike relationship? That would be blue, right? And we can look at P4, and there it is actually yellow. So you can do that for these different locations. And then again, this relationship between the occurrence of the spiking activity and in relation to a particular phase of this beta oscillation, which is shown here in a more handy format, can tell us where the animal attends. So now we have two different codes that could be used here in this area to read it out to a different region and to drive behavior. Now, the next step was for us to see if we can find evidence for these spike phase coding in the human data. And I think I have not shown you that actually the rate is indeed also, and I don't have that slide for some reason here, it must have fallen off at some point, but I can just tell you, you can take my word for it, the high gamma activity is also modulated and gets much stronger, the power of the high gamma activity, when the patient attends to the response field that we are recording from. So there's clearly an effect on the rate here, or what the equivalent would be in this data set as well in posterior parietal cortex. Now, what about the spike phase relationships? Again, I told you that our spiking surrogate is the high gamma activity. And what we did here was that we looked at the phase of oscillatory activity. So here we look at, again, it cuts off at 4 hertz actually to 40 hertz. And we look at the oscillatory activity in this frequency range. And we ask the question, is there a relationship of the high gamma activity consistently to the phase of of any of those oscillations in these different frequency ranges. And what we find is that there is actually a very consistent relationship in this frequency band between 15 and 20 hertz. Again, this is a low beta frequency. In the monkey, it was peaking about 17 hertz. When the animal attends to, when the subject attends to the response field, and this shifts by 180 degrees when the subject attends outward. So again, this is the what you may want to take as the equivalent of a spike phase relationship here. It is a cross-frequency coupling of the high gamma activity to this low beta frequency. And we see that, by the way, if we treat the animal data, the macaque data in the same way, we see a very, very similar relationship. It's a little bit shifted in frequency space, but we also see this reversal here. So now we can ask this question. We have two codes realized in this area. Is one or the other more related to behavioral outcome? And here we just look at the accuracy of the response. So can this code tell us basically if the animal subject or the human subject makes a accurate response or an inaccurate response? And what we see here is that if we look at the spike weight code, there is no predictive power here whatsoever. So that does not inform us of whether an accurate or an inaccurate response will follow. But if we look at the spike phase or the cross-frequency coupling code, then we actually see that we get a better predictive power in this particular example. And it's actually, which is quite nice, on the same order in macaques and in humans. So we think that in this particular example that I give you here, that these spike timing codes that may be implemented in the brain have a closer relationship to drive behavior. This is not to say, because sometimes I think I'm misunderstood on that this is the only code that's used. I think many codes are used simultaneously. Could be that, for instance, the rate code is used for a different purpose here, to be read out for to drive something else. So it's not to say that this tells you anything about the neurocode per se. It can only be interpreted in the framework, I think, of this particular attention task. But it is one of the first examples of a spike phase code that drives cognitive behavior in the primate.
So just to clarify, you showed us all these examples of elevated activity during the delay period in firing rates. So that's apparently orthogonal to this? Because that suggests that the firing rates would predict whether the subject attends or not.
Yes, it tells you whether the subject attends or not, but it doesn't tell you anything about the behavioral outcome. Exactly, exactly, yeah. So just to summarize this part of my talk, I have shown you that these interparietal sulcus areas in the human posterior parietal cortex show spatially specific response fields and attentional response modulation similar to the macaque LIP, and we think that they share a lot of commonalities functionally. We have seen that spatial attention modulates spike rates and high gamma activity, as well as the phase relationship to low frequency oscillations in the primate's posterior parietal cortex. And the behavioral outcomes were better predicted by a spike phase than by a spike rate code in this particular study and in the posterior parietal cortex. Now, we are very, very excited in general about the comparative electrophysiology that we have started. We have just started actually our second task that we study across the primate species, because we think it will offer us a really interesting opportunity to study evolutionary aspects of cognition across primate species. So in this humble beginnings that we are in it right now, we basically look for a lot of commonalities of what is preserved, but eventually, of course, we will hopefully also see a lot of things that are different in these sort of cognitive tasks between the two species. And with that, I thank you for your long attention span. Peter seems to be on the run today. Whenever I see, he's running away from me. So I'm happy to take questions if you have any. Mm-hmm.
So about the first part of your talk, where you showed the beautiful range or causality relationships between this network of cortical areas and the pulvinar, I wondered, since you have spiking activity, I guess the dream vision of what you would like to see, I think, is that you have transfer of information at the spike level between those cortical areas in a way that would depend on whether the pulvinar is synchronizing those areas or not, right? This is how I understood your hypothesis, that if the pulvinar is not synchronizing them, then some information as defined by spiking fails to make it across through the next area. But if pulvinar synchronizes them, then the gate is open and information flows, right? So did you look at that in some way? Because you might have the spikes to do
Yeah,
that.
we looked at spike field coherence. So not in the framework of the Granger analyses, but in general, when we looked at synchrony, we looked at spike field coherence. So we look at the spiking activity in one area and the oscillations in another area, and see if we get an increase in coherence between those two signals, and you get those. So it's not only between the LFPs and between oscillations that you can see these kinds of increases in coherence, but you see that at the spiking level as well. This is how far we have pushed that.
Right, right. Yeah, because you can imagine trying to like do a decoding approach where you say, well, if the pulvinar is synchronizing this, then you can decode, from the more downstream area, as it were, but not if the pulvinar prevents that from occurring or something. That would be really cool.
Sure. I mean, there are a lot of, and there are lots and lots of other aspects we can look in these data. I want to caution it a little bit because you have to keep in mind that these studies are typically based on relatively small amounts of data because this is all done in an awake behaving primate, and you just don't get, really a lot of spiking data at the end of the day. So that limits kind of what you can do with the data quantitatively. And that sounds a little bit funny probably for people who do not do monkey physiology, but it's just a constraint. You can do only so much in a single session, for instance, and so on and so forth. And if you prepare, to record simultaneously from 3 or 4 locations, that constrains you further. You have to match them up and so on and so forth. So in the best of all worlds, yeah, I mean, you would like to have probably data that you could look at in, much more detailed ways, but often you just simply don't have them in these data sets. I think the important things for us to really push the Granger causality hypotheses further is to do causal manipulations, to inactivate regions, to do microstimulation, and so on and so forth, to really assure that we get these causal influences in at least quasi-physiological settings. Many people do not think inactivations are very meaningful interventions, but we can have a discussion about that.
Sort of following up on that, so do we know what happens when people have lesions to pulvinar? Do we see?
Yeah, that's a great question. It's actually quite astonishing. So in the cortex, when you have a lesion within the attention network, very often you get a very severe attentional deficit. So for instance, in parietal cortex, a lesion can lead to what we call visuospatial hemi-neglect, so to the neglect of an entire hemi-space. That's not happening with pulvinar lesions, and that's quite interesting. So you never get a neglect syndrome or anything that comes close to it. And there are a few patients that have been studied by Barbara Fall in Wales, actually, who have relatively small and confined pulvinar lesions. And what you can observe with them is that they basically have mild deficits in visual search tasks. So it may take them double as long to search, but they can search. They can filter, it just takes them longer. They have issues with binding, and those are the typical findings. So they make more errors basically in the binding process. But again, they are not completely unable to do it or at a loss. And we think this fits actually very well with this interpretation we have given our data, that basically what happens under those circumstances is that this cortical attention network cannot work as or operate as efficiently because it's not aligned in time. So what it's probably doing then is it has to really negotiate, just using these cortical pathways, what a good way is for this communication to make it as effective as possible. And that simply takes time. And this is why you see these very, very long reaction times in these patients, in these specific tasks. So they have normal reaction times, if you give them other cognitive tasks that do not engage the pulvinar. So you basically do not render this cortical network completely dysfunctional. You just make it less efficient. And again, inactivation studies would be very useful to probe that in the primate model as well.
So is the idea with the pulvinar that there is like a cortical spatial map that is reproduced in pulvinar to cortex, so that one area maybe alpha oscillations then the pulvinar can tell two certain areas to talk to each other, but a different area in the pulvinar would projected to other locations, for instance.
So the pulvinar, the way you can think about the pulvinar, it's basically made up out of little projection zones that reflect the connectivity, the rich connectivity of the cortex. So if you have, for instance, a V1, V4 direct connection, you will have a little projection zone in the pulvinar that loops indirectly, right? So it's not only, directly direct sequences of regions. It can be, across streams. We have, LIP, V4 connections and so on. There will be a projection zone in the pulvinar for this specific cortical connectivity. So that has been shown for a lot of regions that people claim it's an anatomical rule. I don't know if it has shown for every possible connection that you can think of. So that is how you can think about the pulvinar. Some of these projection zones amount to a map. That is particularly true in the primate for V1 and V2, V4. So there are two maps in the pulvinar that are connected to early visual cortex and then to the ventral stream. The others don't have necessarily a map, but they still have neurons with confined receptive fields, with spatially confined receptive fields. But I think the question that probably maybe a little bit behind, your question is, how do these different regions in the pulvinar know from each other, right? I mean, they have to be coordinated in some way too. And that's a great question because the pulvinar does not have this kind of lateral connectivity that we know from the cortex. So it's not that they can crosstalk so easily. And that is a very puzzling question. And one possibility is that the reticular nucleus of the thalamus basically provides that input because it's like the name says, really widespread a connectivity that includes the pulvinar. And it's also really, really widely interconnected with many cortical regions in the visual system, also in higher order cortex. But that is a puzzle, and we need to solve that by, recording from more regions in the pulvinar simultaneously, and so on and so forth. But that's a question that is very interesting.
Is there ever any reason for surgeons to implant humans with depth electrodes in the thalamus?
It's very rare. I mean, basically, I don't know. You will find one once in a while, but it's very, very rare. It's something that's almost never done. This is one more reason for us to go there as primate physiologists, because the thalamus will be for us in greater parts really uncharted, will remain uncharted if we don't go there in a primate model, because in the human we will probably never learn from these regions, through brain stimulation or these kinds of, recordings.
That was my last little follow-up. As our thalamus brain stimulation experiments done in monkeys?
Not in these regions of the thalamus. They are done, in structures that are related to the motor system, but not in these regions, basically. So the primate model has a lot of value here, very clearly, because especially when it's done in this comparative way, because then we can use it really to understand thalamic function and extend it to the human, at least in approximation. Yeah.
So they, so I would think this mechanism is probably quite generous. It doesn't have to be visual attention. It's auditory attention. It connects to other brain regions. But do you think it may be having different frequency? It's still alpha band, but slightly different. So why exactly, what kind of frequency in alpha band may be specific to the connection to the particular region?
It's a great question. I think there's a lot of confusion in the field what all these different frequency bands mean, and whether also the ranges are meaningful, right? Sometimes you see a peak, at 17 hertz, then at 12 hertz. Is it different, or does it matter? Is this noise? I mean, we just don't know that. For the alpha band, I would think it is very much tied to the visual system. We also see slightly different frequency bands actually in higher order cortex when we look at the frontoparietal network and the dorsal pulvinar that's interconnected with it. because the frontoparietal network uses, beta frequencies, gamma frequencies, so we get into higher frequencies there from the thalamus as well. So it's not a generic frequency, I think. It's a generic frequency for the visual system. We know that for a very long time. I think this is related, to the alpha rhythm that you can see in the EEG, and so on and so forth. This is a very classical, visual rhythm for the visual system. And probably, one of the generators of that rhythm is the pulvinar. But I think the frequency bands of these oscillations depend on the task, on the demands that the task has. I mean, you can think about them as opening time windows. So here we have a 100 millisecond time window that's opened for this network to do whatever it wants to do, right? You can easily see that sometimes you will need shorter time windows. So for instance, if you think about, spatial attention, we know for a very long time that we can move covertly attention every 50, 60 milliseconds, right? And we know from, Tim Bushman and Earl Miller's paper that you see in prefrontal cortex, rhythmic activity around 20 hertz during covert attention. So there you basically have a much, much faster frequency that probably reflects this kind of, covert shift of attention. Just to give you another example. So I think you have to really look very closely what these task demands are. And I think this can, these particular frequencies can depend on many, many different things. The beta frequency that we see in LIP, there is really beautiful modeling work by Nancy Coppell showing that basically these regions are really set up to support working memory networks. So here we are, we are basically tapping into a memory function, and this maintenance activity that we measure there uses also, in other working memory tasks, uses these beta frequencies. So this could be really just a memory network that we are tapping into here. So those are all things that we need to understand. And the only way to understand them is to make links between these oscillations to behavior. And as long as we do not have these links, I think we can only speculate or think they don't mean anything or whatever we want to make out of them. But that will be a really important step for the field, I think, to really establish that that oscillatory activity in the brain relates to behavior. And once you have that, then you can think about how can we now causally manipulate them in order to perturb behavior, right? And we are just not there. This is just not where we are right now. But I think this is where the field has to go. And so we were very happy to see this finding that I showed you of the spike phase code in the second part of my talk, because that's kind of the first little, move in that direction that here, an oscillatory activity can be related to behavior.
How specific is the timing control of the pulvinar in terms of, is basically its role to get kind of these two regions in the room together talking, or does it have a specific effect of selectively syncing, a certain spatial neurons selective to a certain spatial area with, say, neurons selective to barrels? That kind of, is it doing actually causing information to be stored in that manner? Or?
No, I don't think it has. Basically, the way I view it, but again, we know very little about the pulvinar, so take that just in a very tentative way. These neurons are not tuned very well for shape or for orientation or for any thing that you throw at them, basically. They're very sloppy in that. They're visual. You can get the impression that they are visual, but what they are really doing with that visual information, you never get a very clear idea about when you record from these neurons. So the way I view it is that this is really a temporal structure. It doesn't care the information about what the shape is or where to attend to. That's in the cortex. That's the business that the cortex is doing. And the business that the pulvinar is doing is basically just to tell the cortex when it's a good time to align, right? So that this network can talk. It's really a temporal structure. And probably need some kind of very rudimentary content in order to operate that way, but I don't see it, as a structure that would be able to discriminate between these two shape stimuli or anything like it. There has been a literature that is quite old. It was actually done only in one monkey, I think, by Rob Desimone, that has described a filtering function for the thalamus, and that would suggest so that the thalamus basically is able to filter distractor information. And that would suggest that the pulvinar has more specific representations. In our hands thus far, we do not have this impression, and we also do not see these kind of distractor filtering operations in our data set. But again, it's a relatively small data set if you want to look at something like that. So we are not really in a position to make very strong claims about it. But those are all things we need to look at in much, much greater detail as we get more familiar with these structures that have been given up a little bit on for 30 years. So, and again, it's just the very first studies. It will take another few years, I think, until we have a better impression of what's really going on in the pulvinar.
Well, since Peter's abandoned ship,
Yeah.
Thanks, Sabine, again, of course, for the reception up in the center.
Thank you.